the model collaborative research groups in the country, and we're going to hear more about that in his presentation. So David is also the editor of the Red Book, beginning in 2015 and extending to 2024. He's the AAP Red Book liaison to the ACIP. He's an associate editor for the Book of Sarah, and he is past president of the Pediatric Infectious Diseases Society, and I'm proud to say that Chris Bryant is the most immediate past president, and unfortunately she's out of town and wasn't able to join us today. But David is originally from Austin, Texas, did his medical school training at UT Southwestern, stayed there for residency and fellowship, and then came to UAB in 1994 as a virology fellow, and he has been there ever since. I can't really list for you all of the awards and named lectureships that David has had, and he's had over 250 publications. So he's really one of the most recognized names in pediatric infectious diseases. So here's what viral diagnosis and treatment was like in 1983 when I started my internship. Literally doing a viral culture was about the only thing you could do, and it would take about 3 or 4 weeks to get an answer. And it's just remarkable that today we take it for granted that we can make viral diagnoses and that we can treat viruses. That was an unheard of concept back when I started my residency. And to a large extent, this is because of work that David has done with the collaborative group. So his title today is the Odyssey of Therapeutic Discovery, Antiviral Drug Development Programs. I do want to add that our group and others, if you're interested, are going to have a roundtable this afternoon from 12 to 1:30 on blue jeans, where we'll talk about a variety of infectious disease issues. So let me know by email or text if you'd like to join, and I'll send you the link. And David, I do promise you that you can have a rain check on Louisville culture, and we're going to have you come up here physically at some point in the near future. So David, I'll let you take over the slides.
All right, I'll see if I can share. Can you all see the first of my slides?
I can see them. If you want to put them in presentation mode.
Is it, I'm seeing them in presentation mode on my screen. Is that showing up on y'all's?
I see it in editing mode. Julia, can you see the in presentation mode?
No, I'm still seeing it in editing mode. So you are sharing your screen, right, Dr. Kimberlin?
I am. At the bottom it says usbbcollab.com is sharing a window, stop sharing and hide. So I would assume stop sharing means that I am sharing now.
Yes.
And I'm advancing a slide and then going back to my main slide. Is that showing up as movement on y'all's end?
No, it is not. So it's just kind of stuck on editing mode on your first slide. I'm wondering if you go back to sharing the share content portal and choose the specific screen that you want to share if there are multiple options.
All right, I'm back to the main screen. Did y'all lose my slides just now?
Yes, we see you right now.
All right, and I'm having to, sorry, I've got my thing in front of my face, so. All right, I'm resharing. Can y'all see slides?
Yes, they're still in editing mode. So.
All right, I agree. Now I'm going to go to slideshow mode, or is it showing up that way now?
No, we are still in edit mode.
David, if you want to just sort of minimize the slide sorter on the left, and in the meantime, if you want to email me your slides, we might could, you know, I could project them, or
our next best option could be, Dr. Kimberlin, if you share the actual file, your PowerPoint file from that.
I am.
Content.
Oh, do you mean like inside your program?
Yeah, instead of share application screen, you choose share files, and
All right,
you would
I'm
pick your
doing
PowerPoint.
that.
Just a minute to.
And I just need to drag and drop it in.
Yes.
All right, I'm opening. My apologies to everybody. The technology is pretty amazing if you think about it, but also has its limitations.
It does.
All right, dragging and dropping. It says uploading.
Perfect.
Giving grand rounds is a different thing in the COVID world, isn't it?
Now it says converting.
Those are all good signs.
And the little purple circle is going clockwise.
And chief residents have truly become IT professionals.
And my apologies again, guys. I did do the practice session yesterday, and when I could see it as presentation mode, I just assumed the other end of the connection would as well.
And that's okay. Things always happen that we can't predict.
Still converting. Is that okay?
Yeah, we'll give it another minute or so. I think our best last resort can be to have you share your screen and just kind of shrink the left panel of slides.
All right. Well, as it's converting, or at least it appears to be converting on my end, I want to thank everyone for the opportunity to join you today. I do wish it were in person. Obviously, we wouldn't be having these glitches if that was the case. I am honored to be a recipient of this lectureship and to be able to share with you what is truly a group effort. The collaborative antiviral study group has two key words there: group and collaborative. And if you look back, and I'll try to point this out as we move through the accomplishments over these decades of the collaborative antiviral study group, they have consistently included participation from University of Louisville. So this is I'm essentially presenting to you all your own data and the data of many others as well that have contributed to this. And the main reason we need to do it this way is what we study are rare diseases, and if you study a rare disease, then you need to have multiple partners in that, or else it'll take 40 years to get enough subjects at any given study site. So that's the reason we have the kind of structure that we do, and it was really a structure I did not create. Charlie Alford and Rich Whitley had this original concept back in the 1970s. And it was a very prescient kind of a view of things, because it foreshadowed what would happen subsequently with the HIV trial networks, and really now with all clinical research, or at least all substantive clinical research with respect to antimicrobial therapies in pediatrics. So it was a model, I think, that has stood this test of time, and we've certainly adapted as we've gone through as well. Now it stopped doing the little circle, so how do I get to being able to show that, what I just uploaded?
Now you just click on your title there, the antiviral program.
Oh, I see.
And click share now at the bottom.
Share now, got it. Now I just see it on the right side of my screen with a series of small slides. Do I click on one of those? Yeah, I guess so.
There we go. Something's happening on your end. All right, great. We see your first title slide.
Okay.
And you can
if I advance
at the bottom.
Oh, I see. So I click it down at the bottom.
Perfect.
And
yep.
did that advance it? So you see my disclosure slide?
Yes, we do.
Okay. Interrupt me if it glitches, or if I'm not moving through this correctly. Gary, I did silence my phone or put it on airplane mode, so y'all will need to interrupt me verbally if it won't come through in silent mode. All right, in terms of disclosures, the studies that I'm going to be presenting are all NIH funded, and that's the vast majority of the work that I do in terms of research. I do occasionally participate in industry-sponsored studies, most recently Gilead's studies of remdesivir in the pediatric populations. All monies go directly to my university and not to me, and I only have one slide where remdesivir is listed, and we'll get to that in just a moment. In terms of the overall ways by which antimicrobial drugs advance to market, I've illustrated that on this particular slide, where you move from basic research, in this case it would be the development of molecules and the screening of molecules and high throughput kinds of mechanisms to identify what may have activity in vitro against a virus in our case, or bacteria, or fungus, or whatever it may be. And then that moves into preclinical kind of work. And I do have a question for you all right now. Do you see my mouse pointer moving on the screen?
We do not.
Okay, so I will not bother with that. So this is on the left portion of the slide in orange. And then as we move from the preclinical to the clinical area, as you all know well, it moves into phase 1, phase 2, and phase 3 studies. Phase 1 is essentially a dose determining study in the case of a drug. Phase 2 is a more medium-sized kind of study that is a go no go sort of a setup for the sponsor, whether it be NIH or whether it be a pharmaceutical company, to see whether or not they should invest in a large phase 3 study to see whether something is marketable. And then that moves into the full development phase of a phase 3 kind of a trial that hopefully will lead to licensure by the Food and Drug Administration. Once that occurs, you have an approved product on the market, and yet it can still be studied in other populations, or even the population that it was approved in. And that's if that kind of a study occurs, it's a phase 4 study. This is a timeline of, from a historical perspective, of antiviral drugs that have been approved by the U.S. Food and Drug Administration over decades. It starts back in the early 1960s, and this particular slide extends through 2000. The very first antiviral drug to be approved by the FDA was idoxuridine for the treatment of herpes virus infections. It was toxic in terms of oral or systemic administration, but it was approved for topical use for HSV infections. But the first really significant drug that began to change the paradigm that Gary mentioned at the beginning about how unprecedented, unheard of it was to think that you could actually treat a virus, without killing the host cell. The first of those was amantadine, and that was in 1966, I believe. This was an oral drug. We no longer use it because of resistance issues, but an oral drug for influenza A, not influenza B or C, but influenza A only. And then literally, 12, 14 years passed before we started seeing in 1976 what ultimately became what, when I was entering the field, I thought that was the golden era of antiviral drug development, and that was with the drug vidarabine, which is an anti-HSV and anti-VZV drug that was used. It's used topically now, but it was a systemic drug initially, IV, and it really changed the entire field. Because what it did is showed that you could actually treat a virus and kill the virus without killing the host, which obviously would be preferable. And so that was the next major step forward, followed by trifluridine. But then in 1982, the real move forward was with acyclovir. And acyclovir works by taking advantage of a viral enzyme that activates or begins the activation process by monophosphorylating acyclovir to acyclovir monophosphate, that then goes to a di and triphosphate by cellular enzymes and becomes the active acyclovir triphosphate. So if you think about that, the only way that acyclovir becomes active is if HSV enzymes, the thymidine kinase enzyme is in the cell, which means the cell is infected with HSV. And this allows acyclovir to be a very targeted kind of an antiviral drug. And it really showed that, yes, in fact, you could leverage specific aspects of viruses to be able to develop antiviral medications that can target those viruses without, hopefully targeting too much anyway, the host cellular machinery and the host cellular function. And then you move into ribavirin, which was for RSV and Lassa fever, interferon for hepatitis B and C, lamivudine later as well for hepatitis B and C. That's in the green font. But this gray font shows really, if you look to the right-hand portion of this slide, a number of different drugs that were developed for different kinds of human herpes viruses. And then in the very late 1990s, zanamivir and oseltamivir as influenza drugs. And honestly, when I was, arriving in Birmingham at UAB in 1994 and beginning to write studies that were collaboratively joined by other study centers such as University of Louisville, this is what I really thought was going to be the gold standard of when antiviral drug development was really reaching its peak. And then came hepatitis C. And that was a game changer, as they say. And you just can see how much green font there is on this particular slide that goes, all the way on the right-hand side, 25 or 15 more years into the future to 2035. But you can see mostly the drugs under development have been hepatitis C drugs, valganciclovir being the only herpes virus drug until 20 years later. Letermovir was approved in 2017. A couple of influenza drugs, peramivir and baloxavir in blue font, and then the only drug approved so far for coronavirus is remdesivir, being the most recently approved antiviral drug in 2020 for the treatment of SARS-CoV-2, the pandemic virus. This is a map of the sites that are involved, and I did go back and check. If you look for the blue state or the red state rather, in sort of the middle portion on the right-hand side of the map of the United States, that is obviously Kentucky. And I did check to make sure the star didn't shift around, and in fact that's Louisville, and I believe it is. And please don't correct me if I'm wrong on that, because I did double and triple check it, and I hope it didn't get kind of skewed as this thing was uploaded to the website, and so I can project. But you can see we have 35 or so study sites across the United States and Canada. I don't show the Canadian site, in Peru, Lima, Peru, and in the United Kingdom that have contributed to these studies. And I'll mention this, Gary already said, this is historically called the Collaborative Antiviral Study Group. Our current funding is through a U54 mechanism through NCATS and through NIAID, and currently we're called the Congenital and Perinatal Infections Consortium, or CPIC. What I learned is probably don't fuss over what you're called as long as you have the funding to be able to do the studies. And we could not think of an acronym for CASG that would include the word consortium, which is now part of what the RDCRN larger group of consortia that we are fortunate to be part of through this U54 required. So we now are the CPIC. We also go by the CASG. All right, let's talk about neonatal herpes. And this will be the beginning of moving rather rapidly through neonatal HSV, congenital CMV, infantile influenza, and neonatal enteroviral sepsis. This is a series of pictures of babies with neonatal HSV disease. You see on the left-hand panel vesicular lesions on erythematous bases. The vesicles are clumped together, clustered together. It's on the forearm of a baby. In the middle screen, if my pointer would work, I would point to the site of the former scalp electrode. You see some crusting there. And that is essentially a break in the mucocutaneous barrier because of the fetal scalp monitor that allowed HSV exposure to more easily become HSV infection and then subsequently HSV disease. And then the right-hand panel is a baby with disseminated neonatal herpes, DIC, hepatitis, pneumonitis on a ventilator, an older picture, but clearly one that has a dramatic impact. This slide perhaps was reformatted a little bit through the upload, but I do demonstrate on it the 3 different classifications or criteria for both intrapartum and postpartum infection with neonatal herpes. And this is the overwhelming, this 95% of all neonatal herpes is either developed as they are acquired as they pass through the birth canal, an infected birth canal, or following delivery. 85% passage through an infected birth canal, 10% from a caregiver postnatally who kisses the baby, for instance, and has HSV in their mouth. So when a baby acquires neonatal herpes, it will then progress on to neonatal HSV disease. About a quarter of all babies will have the worst form of it, disseminated disease. That's disseminated intravascular coagulopathy, pneumonitis, hepatitis, with or without skin involvement, with or without central nervous system involvement. And the outcomes are worst, the very worst for this particular classification. I'll talk a little bit more about that later on. About a third of babies do not have disseminated disease, but do have central nervous system involvement, and that is called CNS disease. They will oftentimes present with seizures, something very, grossly apparent, but they also can just simply be lethargic or irritable or have some poor feeding. They might have some temperature instability. It certainly could be, group B strep or group B strep or perhaps group B strep or neonatal herpes, which is much less common. But it can be, a number of other things as well. It could be infantile histoplasmosis in our parts of the country, for example. And then finally, the remainder, 45% or so, they don't have disseminated disease. It's localized. It does not include the central nervous system. So it's called skin, eye, or mouth disease, SEM disease. This has the best outcome as long as it does not progress to either CNS disease or to disseminated disease. On this slide, I give a historical perspective of what we accomplished from the 1970s through the 1980s with respect to treating neonatal herpes. And this was from 74 to 91. The bars are clustered under disseminated, the 3 bars to the left, CNS, the 3 bars in the middle, and skin, eye, mouth, the 3 bars on the right. You want to have more of the purple and the yellow in the bars where they have normal development and are alive or development unknown. You do not want to have red in the bars, and you would prefer, if you're going to have red initially, to have more green than red, because that at least means they've survived. And what this shows is that the initial studies that Rich Whitley led in the 1970s of vidarabine versus placebo, it was ethical to do a placebo trial at that point because we had no antivirals that could work, that vidarabine treatment decreased mortality for both disseminated disease and CNS disease. And if my pointer worked, I would point out the middle bar of the cluster of bar graphs for disseminated and the middle bar of the cluster of bar graphs with CNS to demonstrate that. So we had less death by using vidarabine. That quickly became standard of care. And then when acyclovir became available, there was a phase 3 study conducted during the 1980s of vidarabine versus what we would now call standard dose or lower dose acyclovir, 30 milligram per kilogram per day of IV acyclovir. And interestingly, this was the only trial of a head-to-head comparison of vidarabine and acyclovir that did not show superiority of acyclovir, unlike herpes encephalitis in older people and multitude of other studies of herpes infections in other populations. There was not a superiority with acyclovir, but it was much better tolerated than vidarabine. And so it then became standard of care for neonatal herpes. The standard duration of therapy was 10 days. And again, the dose of acyclovir being used was 30 per kilo per day. And I say that for a purpose. Now, let me tell you a little about a patient that I was involved with the care of many years ago. This was a baby who was born in Montgomery, Alabama, about an hour and a half south of Birmingham. And this was in the pre-digital MRI and pre-digital CT era. So this was a picture of a literal film that, you know, we used to put up on the boards. This baby was born at 34 weeks, and then at 10 days or 11 days of life, developed a vesicular rash on their right clavicle and on their right scapula. And the neonatologist looked at it and said, well, that may be herpetic. They started acyclovir at 30 milligram per kilogram per day. They did not do an LP. The following day, the baby had a seizure. So they did a lumbar puncture. It had 33 white blood cells and a protein of 143. And they had already sent off, before they started acyclovir, they sent off swabs of the lesions for HSV culture. When they did the LP the next day, they sent off the CSF for HSV PCR, and they maintained acyclovir at 30 per kilo per day, the lower dose of acyclovir. This was in the early 2000s. So at that point in time, the neonatology service kind of changed over. A different neonatologist came in a few days later. The lesions were gone. That neonatologist looked at the results. The CSF PCR was negative. The HSV culture from the skin lesions was negative. So on day 8 of therapy, they stopped acyclovir. And baby was about 21 days old at that point in time. They went home. And then at 2.5 months of age, the baby presented to a local doc in the box with a vesicular rash on their thumb. That's when they called me. I said, send the baby up to Birmingham. Let's have a look. Baby came up. They did look herpetic on the thumb. I unroofed one of them, sent that for culture, and it grew HSV type 2, which really, the only way you can really link that in with the history I've already given is that this baby, in fact, really did have neonatal herpes and had gotten what would have been standard of care in the 1980s, 30 per kilo per day for 8 days, whereas standard of care was 30 per kilo per day for 10 days at that particular time in the 1980s. So we got an MRI of the baby's head, and this is what we saw. And I say this to point out that this is a rationale for a study we did in the 1990s of higher dose acyclovir for longer durations of time. And cutting to that particular slide or first of slides of a Kaplan-Meier plot of survival, where you have proportion surviving on the y-axis. You want to be higher up on that y-axis. That means you're more likely to be surviving. And time is on the x-axis. And what you can see is that the green lines represent the survival of babies receiving higher dose acyclovir with disseminated disease, receiving higher dose acyclovir, 60 per kilo per day. That was what was being studied for a longer duration of time, 21 days. And treating with higher dose acyclovir, 21 days, mortality from disseminated disease is 30%, survival is 70%. Compared with what we had seen in the 1980s, the lower dose acyclovir for 10 days, survival was 40%, mortality was 60%. So we were statistically significantly more likely to survive disseminated neonatal infection by increasing the dose and treating for a longer period of time. Now, the same Kaplan-Meier plot for babies with CNS disease shows a similar separation of the lines. This was not statistically significant because more babies survived neonatal herpes when they had CNS disease, and they were treated with 30 per kilo for 10 days. About 20% died, 80% survived, whereas with higher dose acyclovir, 95% survive, 5% die. So you see the same kind of separation of the lines as you move left to right across the screen, but the amount of disease death in CNS babies was simply lower, and that, in my judgment, is why there was not a statistically significant difference. On this slide, I show the morbidity among survivors, and this is morbidity at 12 months. There are 3 clusters of graphs here, of bars here. The left-hand 2 bars are for skin, eye, mouth disease, The far left is the babies with skin eye mouth disease receiving 30 milligram per kilogram for 10 days. Immediately to the right of that is 60 milligram per kilogram for 21 days. Then in the middle two bars are the CNS babies, 30 per kilo per day, 60 per kilo per day. And on the right-hand two bars, 30 per kilo per day for disseminated, 60 per kilo per day, far right. And what you can see is a couple of things. Number one, far left bars, all the babies with skin eye mouth disease do well. They don't have brain involvement, so you would expect that their morbidity to be nil, and that's what it is. The far right bars show that with 30 per kilo per day with disseminated disease, about 60% of babies have normal development, whereas the far right column shows that with 60 per kilo per day for 21 days, we can increase that, the portion of that bar that's yellow, up to 85%. That's good. That means you're more likely to survive and you're more likely to be normal if you have disseminated disease. But the two middle bars are CNS disease babies, 30 per kilo per day and 60 per kilo per day. And look at the proportion of those two columns that are yellow. It's about 29% in the 30 per kilo per day column, and it's 31% in the 60 per kilo per day column. No difference whatsoever. And I want you to keep in your mind what those columns look like, because I'm going to show you again. I'm going to show those to you again here in about 2 or 3 slides. But I just memorize or keep in your short-term memory here what these two middle columns are looking like. So what this led us to wonder or postulate or hypothesize is whether or not giving oral acyclovir suppression could increase the likelihood of better outcomes among survivors, especially of babies with CNS disease, because we were not getting that kind of improvement from IV acyclovir, higher dose, longer duration by itself. I will mention that, again, University of Louisville participated in these studies that are represented on this slide and the prior two Kaplan-Meier curve slides, as well as the studies that I'm showing now, or that I will be showing now. So what the next study we did was, and this was in the 2000s, we designed a study where babies completed IV acyclovir, higher dose, 21 days for CNS, 14 days for skin eye mouth. And then they were either randomized to immediate oral acyclovir suppressive therapy. We'd already done an earlier study to determine the dose. This was a phase 3 study I'm showing you here. Or they were randomized to placebo. And if they had a number of skin recurrences thereafter, those skin recurrences, of course, were treated episodically for X number of days until the skin lesions resolved. They went back on the placebo. It was blinded, but that group was on placebo. But if they had a number of these recurrences, the study drug was allowed to be stopped, and they were put on open label oral acyclovir. So we had a group that immediately started suppressive therapy, and we had a group that did not start suppressive therapy unless they had a number of skin recurrences, which most did, and then they had a delayed onset of oral acyclovir suppression. It took 11 years to do this study. These are difficult trials to do, and this is the end result of what we found out. I want you to look at the middle columns here, the second from the left and the middle column. That's for the 103 study, the CNS baby study. And you can see that the group receiving acyclovir had a median Bayley mental score of 90.5 versus the group getting placebo had a median Bayley score of 66.5. And with Bayley scores, you want to have a higher number. That means you're more likely to be normal. And this difference was statistically significant. Now, we in the far right columns, we did not see a difference in terms of developmental outcome differences with skin eye mouth, but that's to be expected because they didn't have CNS involvement. They didn't have brain injury to begin with, in other words. So this study showed us that oral acyclovir could improve those outcomes. Now, we also broke this down categorically. The Bayley scale is a continuous scale. You can score an 88 or an 89 or a 90 or a 91, for example, but you can group those into normal development, mild impairment, moderate impairment, and severe impairment. And that's what's represented with these two column graphs. The left column graph you can see right now shows the babies that were randomized to placebo following receiving 60 per kilo of IV acyclovir for 21 days. And then the right-hand column is the group that was randomized to immediate suppressive therapy. And you see that about 30% of babies randomized to placebo were developing normally at a year, whereas it was about 70% if they were put on immediate suppression. And I'm going to show you those two graphs I asked you, or those two columns I asked you to hold in your short-term memory here. And I think it's just remarkable how similar the column, the column for the placebo of the group in the oral suppression study, how similar that looks to the two graphs, the two columns that I put up now on the left-hand portion of the screen. And it illustrates to me that this really is a treatment effect. And as well, with the New England Journal paper, and I don't know exactly what the schematic is here, what that symbol is, but in any case, what you can see is the median score. The top, second from the top column is the median Bayley score for people that were on active suppression from the very beginning, randomized to oral therapy. The bottom row are those that never got active suppression because they didn't have skin recurrences. And the middle row are those that were initially put on placebo and then had a few skin recurrences after 2 or 3 months. And so they ended up getting suppression that was delayed in onset. And you can simply look, down that middle column. The numbers are higher for the group getting immediate suppression. They're lower for the group getting no suppression. And it's moderately in between for those that had deferred onset of suppressive therapy. Again, showing a treatment effect if you are immediately started on suppressive therapy. For the sake of time, I'm going to move on to the congenital CMV discussion. This is a picture that was taken by Sergio Stagno, who Dr. Marshall mentioned. He was one of my mentors, chair of our department for 25 years. This was a picture taken in the 1970s when he was an active researcher in the congenital CMV field. It's of a baby with symptomatic congenital CMV infection. You might appreciate a microcephalic appearance to this baby's head, a ruddy kind of a hyperbilirubinemia, and a petechial rash. Now, the following image is not this particular baby's MRI, but it likely would be what this baby's MRI would have looked like. Periventricular calcifications, I should say CT scan, not MRI, seen on this particular image. Why do we care about congenital CMV? Well, number one, it is the most common congenital infection, most common congenitally acquired infection in the United States. About 0.5% of babies that are born in the U.S. will have congenital CMV. That equates to about 20,000 babies a year, 20,000 babies a year. And it's the leading infectious cause of deafness. And what the left-hand pie graph shows are causes of deafness at birth. The red portions of the pie graph are the congenital CMV slices of this, asymptomatic at birth and symptomatic at birth. We'll talk a little bit more of that over time. And then at 4 years of age, the right-hand pie graph, and I apologize for the shift as this uploaded into the system, but the pie graph goes from about 20% at birth to about 25% at 4 years of all causes of deafness being related to congenital CMV. This is a big deal. Now, if you look at the characteristics of the patients who have audiologic sequelae from congenital CMV, there is a difference in likelihood based upon whether the baby is symptomatic at birth, whether you can walk across the nursery and look over and say, there's something wrong with that baby. I need to evaluate that baby for X, Y, and Z, and X, Y, or Z is CMV. In other words, there's a clinical reason for you to be thinking about congenital CMV infection. And that would be a symptomatic baby versus the more likely scenario. About 90% of all babies with congenital CMV have no outward sequelae or visible manifestations, I should say, at all. They are asymptomatic congenital CMV infections. And you can see that about 40% to 60%, in this slide it's represented at 41% of babies with symptomatic infection will have sensory neural hearing loss at some point. Of the asymptomatics, it's between 5 and 10%. In this particular study, it was 7.4%. But the important things that I want you to look at are in the far left column. This hearing loss can be unilateral, it can be bilateral, it can be high frequency only, it can be delayed in onset, it does not have to be present at birth. It can be progressive, it can fluctuate over time, it can get better and get worse and get better and get worse, or it can simply get better. And that's without any treatment whatsoever. So it's very important that we design studies of the treatment, evaluating treatment for congenital CMV that include a comparator, because if it can get better on its own and you treat everybody and it gets better, you might erroneously think it's the antiviral drug that's doing that when in fact it's not. Now, I want to talk briefly about the universe of babies that are destined to have sensory neural hearing loss. So it's the 41% of symptomatics and the 7.4% of asymptomatics. And on this slide, I show when those babies cumulatively acquire hearing loss. You can see that about 45% of symptomatics will have hearing loss at birth, 55% will not. About 25% of asymptomatics will have hearing loss at birth, about 75% will not. And the likelihood just steadily increases over the first 5, 6, 7 years of life. And I point this out simply to say that if you rely on babies who failed their newborn hearing screen to be the only ones tested for congenital CMV, you by definition are going to miss a majority of them. Because anywhere from 75% of asymptomatics to 55% of symptomatics will not, who are destined to have hearing loss, who are going to have hearing loss at some point, do not have hearing loss at birth. Now, the first study of treating congenital CMV was done in the 1980s. Again, Rich Whitley and Charlie Alford went to Sandoz, I believe, and secured intravenous ganciclovir, which was a study drug at that point, for the study of it in babies with congenital CMV. They studied 2 doses, a lower dose of 8 per kilo per day, a higher dose of 12 per kilo per day. The y-axis here is the amount of virus in the urine. It was by quantitative culture. And the x-axis is study day. And they chose a study duration of 6 weeks, not because of some animal model that suggested that was going to be the best time duration, but simply because this drug has a lot of toxicity. And because of that, they felt like they could only treat for 6 weeks. And they showed that they could clear the virus from the urine after 6 weeks of therapy. But like all human herpes viruses, the virus did rebound once antiviral therapy was stopped. So we then took that in the 1990s, and with the assistance of colleagues at University of Louisville and others around the country, we evaluated 6 weeks of intravenous ganciclovir therapy versus a randomized no treatment group. And what I show on this slide is 2 pie graphs. The pie graph on the left is the hearing outcomes at 6 months for babies who had ganciclovir therapy. The pie graph on the right is the same for the babies in the no treatment group. The red slice of the pie graphs are those who had worsening of their hearing, or if their hearing started off, moderately impaired, it remained moderately impaired, didn't get better, in other words. And you can see that the ganciclovir group, about 31% of ears had worsening or maintained the same degree of abnormality versus 61% of ears in the no treatment group, and this was statistically significantly different. Looking at it at about 2 years of age, same groups, you can see that 52% of ears in the ganciclovir group had worsening or remained abnormal versus 78% of ears in the no treatment group. Again, statistically significantly different, showing an improved outcome with antiviral therapy. So then the question was, well, what if we could treat longer? And around that time, valganciclovir was becoming available. We did a study that, again, University of Louisville was involved with, called the CASG 109 study, where we determined in a PK/PD pharmacokinetic pharmacodynamic study what the dose of ganciclovir, oral valganciclovir, should be that gives the same systemic exposures to ganciclovir. Once we had determined that dose to be 16 per kilo per dose, given twice a day orally, because valganciclovir can be given orally, we then conducted a study of 6 weeks versus 6 months of therapy. This was a placebo-controlled blinded study. The 6-week outcomes at 12 months are shown on the left pie graph. The 6-month treatment outcomes at 12 months are shown in the right. And you can see that there is less red with longer treatment, that hearing outcomes are better when you treat for 6 months compared with if you treat for 6 weeks. And a similar kind of finding was seen out at 24 months as well. It's not a huge effect, but it is a modest effect. And then we also saw, and I apologize for moving quickly through this, I want us to cover as much of these materials as possible. We also saw an improvement in Bayley developmental scores when babies were treated for 6 months versus 6 weeks. And the portion, Bayley 3, you may know, has 7 components to it. So we did a Bonferroni adjustment so that we wouldn't have multiple testing kinds of biases. And the language composite score and the receptive communication score were statistically significant even following that Bonferroni adjustment, where you had higher numbers, better outcomes with the Bayley in the 6-month treated group as compared with the 6-week treatment group. And as Jim Cherry out at UCLA pointed out, all of the column, or rows, I guess, are higher in the 6-month column than the 6-week column, with what otherwise would be statistically significant, except for the fine motor scale, if we had not done the Bonferroni adjustment, suggesting that, yes, in fact, we do see those kinds of better outcomes. Now, all of these studies that I've talked about so far with congenital CMV has started therapy in the first month of life. We have just completed a study with partners in the United Kingdom of starting therapy beyond the first month of life in babies with congenital CMV associated hearing loss. And I'm going to show you a number of slides here rather quickly. This is called the Valgan toddler study. The take home here is it does not work when started beyond a month of life. We had 6 weeks of valganciclovir therapy in the second column from the left. The third column from the left is 6 weeks of placebo therapy. The rows are improved hearing at 6 months, no change normal to normal hearing between baseline and 6 months, no change abnormal, like mild at birth and mild at 6 months, or worsening. The comparison for the primary endpoint was the proportion worsened versus everything else, and it was not statistically significantly different. If anything, more of the Valgan treated group worsened compared with the placebo group. And when we did other comparisons of improved versus others, nobody improved. There was no difference whatsoever. When we looked at improved or normal to normal, no difference, p-value 0.48. When we looked at best ear hearing, not total ear hearing, we can discuss that in the question and answer if we have time in a bit. Again, no difference. If anything, the Valgan group randomized to valganciclovir actually did worse than the placebo group, and no difference here when we do it improved with best ear, improved, no change, normal, normal, or and when we look at the post hoc analysis of those that were asymptomatic or symptomatic at birth, just kind of, we didn't do statistical analysis here because the numbers were so small, but you just kind of scan these columns, and the columns in this kind of orange color all look about the same. The columns in green look about the same. We just simply did not, this is about the most negative study I've ever been involved with. And then we did a similar kind of thing for those that were started in a treatment beyond 1 month of age, but within the first year, versus those that were started between year 1 and year 4 of life. So we just did not see anything. There's a group in the Netherlands that had a study design. It's called the Concert study. Study design was less robust than us. It was an open label study. They do have some interesting statistical analyses that show a difference. They're going to be presenting those data today in Rome, Italy. But I I don't know. I would not start antiviral therapy beyond the first month of life. All right, influenza treatment. We know it's bad. It wasn't bad last summer, or last winter rather, for reasons we're scratching our head about, but probably it was because of masking. But typically somewhere between 150 and 200 children die every year of influenza. Prior to our study of Oseltamivir in children under a year of age, the approved indication for Oseltamivir was 1 year of age and older, but we did a study of Oseltamivir carboxylate. The concentration of the active metabolite of Oseltamivir is represented on the y-axis. The study groups are on the x-axis. We had, starting with the far right, babies that were 0 through 2 months of age, then immediately to the left of that, babies who were 3 through 5 months of age, then to the left of that, 6 through 8 months of age. You can work your way across. And we studied different dosages of Oseltamivir in this population, and we had a target range pre-identified. Those are the upper and lower lines that are in purple, and we had a target that's in blue. And you can see that we hit the mark pretty well with an adjustment for the 9 through 11 month old. That's the middle column, where we did have to go up a bit in the dose to be able to get it consistently between the purple lines. And we were able to identify what that dose is. And we did so during the time that the last pandemic, the 2009-2010 influenza pandemic, was ongoing. We also did a study that was only done at one site in North Carolina of premature babies that are represented in the green dots that were treated with a dose that we anticipated, hypothesized was the correct dose, and it turned out to be relatively accurate. Now, I'm moving through this quickly because on, I believe it was Sunday, April 26, 2010, I was in the office, I was on service, and I got a call from the Food and Drug Administration. This study was ongoing at that time, and they said, look, y'all have the only data in the world on treating babies with Oseltamivir under a year of age. We would like for you to release that partially completed data set to us. I immediately called the NIH on that. They said yes. By the following day, we had released the data set as it was at that time, that was summarized more completely on the prior slides, to the FDA, and they had issued an emergency use authorization for Oseltamivir. It was one of the more gratifying parts of any of the studies that I've been involved with, because it immediately had an impact on the care of babies worldwide. That's the kind of impact that these studies, the scope of these studies, can potentially have. We were in the right place at the right time, and we were privileged to be able to contribute to that. And ultimately, it has led to recommendations by the American Academy of Pediatrics and the Centers for Disease Control and Prevention and also FDA for licensure in term babies, for how to dose Oseltamivir across the ages up to 2 years of age. Briefly, let's talk about neonatal enteroviral sepsis. This is a study we did of pleconaril, which is a drug shown in a very large schematic on the left portion of this that fits into a lock in the middle portion of the slide that is part of the outer coating of an enterovirus, and it essentially locks up the enterovirus so it cannot uncoat when it infects a cell, and if it cannot uncoat when it infects a cell, it can't replicate in that cell. So it's an anti-enteroviral drug. It's not really, it's kind of hung up right now. Ours was the study that showed benefit, and I'm afraid this may not show well. I apologize, this is the conversion issue. But what we found is that time to culture negativity was almost statistically significantly better. In other words, babies treated with pleconaril with neonatal enteroviral sepsis cleared their enterovirus more rapidly than did those treated with placebo. This was a study that we conducted, but it was led scientifically by Mark Abzug at the University of Colorado. But when we looked more deeply at, this was a better slide that didn't get so corrupted, 2-month survival, where you want to be on the y-axis again, higher up. you can see that survival was improved among those with enteroviral sepsis in the intent to treat analysis compared with those getting placebo. And we saw something similar with the intent to treat analysis with 2-year survival as well. Now, what we did not see, though, was for those that were proven to have enteroviral infection, Enterovirus-Confirmed disease, we did not see a statistically significant difference, although the lines do diverge in the same way and the numbers are smaller. And so we're working with the FDA now with on behalf of a small company that owns this drug. We're getting no compensation, obviously, from that. To see whether these data might be robust enough to have a pathway toward some sort of limited approval for neonatal enteroviral sepsis. We are not there yet. Final slide. What are we doing now? Well, these are the current CPIC and CASG studies. We have a natural history study. This is of acute flaccid myelitis. This AFM study is the NIH's major investment in this emerging infectious diseases. It's related to Enterovirus D68. The asterisk indicates that U of L is involved with that study. We have a study that will be starting very soon of trying to establish a biomarker in congenital CMV infection. We're going to be screening babies for asymptomatic infection and then doing a number of different blood and imaging testing to see what we can identify as a biomarker to predict who is more likely to have sensory neural hearing loss. That will make it easier to develop subsequent trials of new antiviral drugs. We have a natural history study for enteroviral sepsis, same kind of goal to identify endpoints that could help in the development of future studies of anti-enteroviral drugs in this very much needed population. We have a Valaciclovir PKPD study, pharmacokinetic pharmacodynamic study in neonates. Again, all these asterisks indicate U of L is involved with this. We have a Letermovir PKPD study in congenital CMV, also with U of L involvement. We're following up those 6-week versus 6-month CASG 112 study subjects with congenital CMV. They're about 12 or 13 years of age now. We want to see if the differences in hearing outcome maintained or not. And then we have an incidence study for HSV in Peru. So with that, I thank you very much for your attention. I turn it back over. I think we're at top of the hour. Gary, I don't know if we have time for questions or not. I apologize for the rapid sort of move through here, but I think what I attempted to do with this was to show more the flow of how things move through the overall system of proving drugs or disproving drugs to work in the neonatal population, more than each individual disease being drilled into. So I hope it was with some degree of success to see how working together we can accomplish a lot.
David, thank you so much. It's very clear to me that you've upheld the standard of the Perlstein lecturers over these last couple of decades. We are at the top of the hour, and I want to invite folks in the audience who have questions for David. If you could join us at noon for our little virtual roundtable, you could feel free to ask those questions then in person, or you could email me the question and we'll ask David and we'll get an answer to you. I just want to ask you, David, maybe in the next few minutes before we close, could you say something about the structure and organization and governance of the CASG? You know, we have a lot of junior faculty members who might be interested in becoming part of collaborative networks like that. So how is it done, and how are decisions made, and how do you get involved?
Yeah, that's an excellent question, Gary, and I appreciate very much you giving me the chance to address it. That's really one of the benefits of this new funding stream, this U54 that we have for what we now are calling the CPIC studies, which were the majority of those studies that were on that final slide that I showed. And what it means is that in this case, it's Chris Bryant who's involved as the site PI, but this is as part of a larger rare diseases research network consortium, the RDCRN. One of the real goals of that NIH pipeline is to get junior faculty and fellows involved in the processes. And so by being a site within the CPIC, the consortium that we do, Chris can then open up the door to junior faculty and fellows to become scholars in that program, and then they get to not only have the benefit of actually learning about doing rare diseases research, but they get to put that on their CV that they were an NIH scholar. That carries a lot of weight, in terms of promotion or getting your first job in the first place, and you learn a lot as well. And then also to compete for pilot funding, which is a substantial amount of money that we have through the CPIC, again, part of the RDCRN. And by doing it through CPIC, they also then can get access to other consortia. There are like 20 consortia within the RDCRN. And I don't know if Louisville is part of those other consortium or not. But if you're a geneticist, for instance, you can be in, or you study porphyria, and you're not part of that particular consortium, through the CPIC, you can get access to that consortium and get in more with people that are studying the kinds of diseases that you like to do. And we would love, if y'all get to do that, we win. So, it's an across the board win. When we recompete, we say, look how we helped bring the next generation of folks into this particular field. And so please talk to Chris. She can talk to us. We can get you involved. You do not have to be an infectious disease doctor to do this. You can be a neonatologist. You can be, a geneticist. You can be a pulmonary doc. You can be any of the subspecialties and be part of this.
That's great. Well, thank you, David. And again, it's great to have had you. And we'll see you at 12 Eastern, 11 your time.
Look forward to it. And this time, hopefully, I don't think I have any slides, so hopefully we won't have these kinds of glitches.
We'll be on blue jeans. It'll be easier.
I do thank everybody for the opportunity to be with you today. I apologize for running over. Thank you for what you do. And I do hope that I get up there to be able to see you in person and shake hands without a mask at some point in the future. We'll see. We'll see how that unfolds. It won't be in the near future, but one day. Thank you very much.
Great. Thanks. Bye-bye, everyone.
Thanks so much, everyone. Have a great morning.